Okay, Dr. Johnson, could you describe what the Marvin Mouse is and what it does for your research?
The Marvin Mouse is essentially a project that was funded by the Marvin Lewis Foundation, and it was essentially an idea we had about 3 years ago to try to understand better how is the immune system involved in multiple sclerosis as well as related diseases. The Marvin Lewis Foundation has had an interest in multiple sclerosis since Marvin's arrival in Cincinnati, partly due to the fact that his brother-in-law had MS, and he in particular was interested in funding projects related to this. We came to them with essentially the blueprints for the genetic project, which was essentially to try to create what's referred to as a transgenic mouse, which would enable us to study a particular type of immune cell which is very important to multiple sclerosis research, and that is the killer T cell. In about the last 10 years or so, the killer T cell has become very important in multiple sclerosis research because it is found in multiple sclerosis lesions in high numbers and is located in areas of tissue damage. For example, there's a lot of sick and dying neurons in close proximity to these killer T cells that have entered the central nervous system. Now, the fact that they had entered the CNS and had been next to these sick neurons is not enough in these particular studies to determine if the killer T cells have the capacity to damage or hurt these neurons. And so the idea was to try to create the Marvin mouse, which is essentially a transgenic mouse, which would enable us to take away the ability of killer T cells when they enter the central nervous system to engage neurons specifically. And the reason for doing this is to try to create a proof of principle project which would enable us to at least determine, in medicine at least, if the killer T cell has the capacity to engage a neuron in our models of multiple sclerosis, and the mouse being the one that we are currently using. The central purpose of this is once this is established in medicine as a potential mechanism by which a killer T cell could contribute to pathology in the central nervous system, we now could design therapy regimens designed to block such interactions therapeutically in human multiple sclerosis. And I think the reason for this is essentially this type of interaction will not be taken serious in human medicine unless it is essentially shown scientifically to be relevant in models such as the one that we just created.